Hello, this video is intended for RCBC students of general biology, but we hope others will join us. We're picking up on our discussion of HIV, and specifically we're going to talk about treatments that we have to keep viral titers low. One of the challenges with HIV is its ability to mutate. The HIV that you see today is a little bit different than the one that we may have seen 5 or 10 years ago. And the reason is the enzyme here, reverse transcriptase. This is the enzyme that takes the viral RNA and converts it into DNA so that it can integrate into our host genome, our chromosomes. It is a form of polymerase, and as we discussed before, polymerase has the ability to check for mistakes and correct those mistakes. However, this reverse transcriptase doesn't have that proofreading ability. So when it makes copies of the DNA, it makes mistakes. Therefore, those mistakes are built into the new viruses that leave the cell. And so each virus is going to be a little bit different. Let's talk about some specific antiviral drugs specifically for HIV. These drugs have helped to decrease the mortality rate of HIV. They stop viruses from growing or proliferating. Now, I mentioned earlier keeping the viral titer low. That's what it means, keeping the number of viruses low. It's not getting rid of the viruses altogether. Now, there are multiple strategies, and we'll talk about ones like AZT, also protease inhibitors. And the thing that I want to mention in this is about the cocktail. Often when people are HIV positive, they are taking multiple medications. That's what we're referring to in a cocktail. They take them together so that they can kind of chip away at HIV from multiple different angles. And that helps with this highly mutable virus, because if it mutates to avoid one medication, maybe the other two will continue to work. Let's review the life cycle of HIV, because that's how we're going to approach crippling its ability to make copies of itself. Remember, the virus needs to dock at a CD4 receptor. Then the genetic material will get dumped into the cell. That genetic material is RNA, so we need to make a copy of DNA that requires reverse transcriptase. Then the DNA will move into the nucleus and needs to integrate, kind of slide its way into our chromosomes. And there's an enzyme called integrase that is required for that process. Once we stimulate this T cell to begin growing, so do the viruses grow. And you can see that we're going to go through transcription. We're going to make new RNA, and we're going to make new proteins. Those are going to assemble together, and then they are going to bud off, and new viruses are going to be formed. The last step here, which you can't see in the picture probably because my face is in the way, is requires something called protease. When we make proteins, we make one mRNA strand, and then one protein results from that one mRNA. When viruses do it, they make a long transcript with multiple proteins on it, or multiple mRNAs to make a protein. And then that protein, once it's made, needs to be chopped up so that it can become functional. And protease is an enzyme which breaks down proteins. And in particular, there's a specific protease that breaks down the proteins of the HIV virus. So this is what we're going to do, is we're going to take a look at this life cycle, and we're going to try to inhibit different processes. We can inhibit this interaction with the host. We can inhibit this creation of DNA from the RNA. We can inhibit the integrase. We can inhibit the protease. But let me tell you about one story which talks about how we might inhibit the entry. Don't even let the virus get in. This is J OBrien. J OBrien started to study HIV years ago. And one of the things that he noticed is that we know there are certain risk factors which increase your likelihood of becoming infected with HIV. And he noticed that there were individuals that were in that group. Those risk factors pertain to them, but they never seemed to be HIV positive. And he started to investigate. There's a great book called Tears of a Cheetah, but he started to investigate, and he found that there was actually a broken gene in those individuals, or a broken protein in those individuals, that prevented the virus from getting into the cells. So the risky behavior may have brought HIV into their body, but the HIV could not get into the host T cell. It's also very interesting because he started to do something called molecular clocking, which allowed him to figure out when this mutation arose. How long ago was this mutation introduced into the human population? And he figured that the way evolution worked, it couldn't be HIV that was selecting for that broken protein, that it must have been something else that happened years and years ago, in fact, hundreds of years ago. And he then was able to determine that it was something that happened approximately, say, 600 years ago. And that was the bubonic plague. That there were individuals who survived the bubonic plague when everyone else was dying. And as it turns out, the mechanism for how Yersinia pestis does damage to the human body is the same mechanism that HIV uses to get into the cell, so it can do its damage. And we will talk about one of the reasons why there is such a high incidence of HIV in sub-Saharan Africa towards the end of this lecture. So let's take a look at what J OBrien figured out. He and other scientists, here is the virus here, and here is GP120. And then this is the T cell. And the T cell has this CD4 on it. I mentioned this one to you before. But there's also what we call a co-receptor, CCR5. And when HIV comes in and attaches itself to CD4, the CD4 will actually bend, and the GP120 is looking for this CCR5. It's only when HIV can interact with both of those proteins, both CD4 and CCR5, that the virus is able to get into the cell. For those who seem to be immune to HIV, what they don't make is this receptor right here, this CCR5. So the CD4 interacts with the virus, the CD4 bends down, but the virus never can find that CCR5. So this fusion event does not happen if you do not have this CCR5. So people who are immune to HIV have a mutated gene which doesn't allow this protein to be made, certainly not made in its functional structure. And that mutation arose hundreds of years ago because certain people had a broken gene which made them selected by nature due to the bubonic plague. Now, you can take basic discovery and then turn it into technology. There is a drug called Maravirac, which has been created, which sits down on the CCR5. And so even if you have good CCR5, when the virus is trying to get into your cells, the Maravirac blocks its entry by not allowing the GP120 to interact with the CCR5. So that's one drug that blocks viral entry. Another drug that was created years ago by GSK is called Retrovir, or AZT. AZT prevents the reverse transcriptase from working. it prevents the DNA from being created from the template of the viral RNA. Now, we discussed earlier on how in replication, in the synthesis of DNA, that they needed to have a free 3' hydroxyl group. At the time, you were probably thinking like, boring, who cares, right? What does that mean? And I will show you now. We'll take a look at this over here, which is thymine or thymidine. And this is the sugar. And remember, we've talked about this hydroxyl group, which is attached to the 3' carbon. When DNA polymerase sees this base, this nucleotide, and it wants to bring another nucleotide in, it's looking for OH. It can't bring it up over here. Remember, it can only move in a 5' to 3' direction. And the reason is that this 3' hydroxyl group is necessary. So what AZT looks like, it's called an analog. An analog is something that sort of looks like something else. So this sort of looks like thymidine, but over here you can see we have changed the sugar so it no longer has a hydroxyl group. So when the viral DNA chain is trying to be created and a new nucleotide comes in after AZT, the nucleotide has no hydroxyl group to hook to, and therefore the viral copy DNA is not created. This AZT actually is a competitive inhibitor, and in the polymerase, in the reverse transcriptase, it won't allow the normal process to happen. So when we talked about inhibitors and being competitive because you're in the active site, that's what's happening here. The last thing that I want to tell you about is a protease inhibitor. Similar idea, right? This is an enzyme. This is the active site. This is where that string of connected proteins would bind and get in here. And then this enzyme, which is in green, would clip those proteins so that you could have a functional virus. If you have an inhibitor to that protein, in this case, the inhibitor gets right into the active site. It's a competitive inhibitor, and it will no longer allow the viral protein connected strand to get into where it needs to get to, and therefore no viable virus is made because the protein cannot be cut. These are some approaches. As we said, we take multiple of these medications, put them together in a cocktail in order to keep the viral titer low. We don't get rid of the virus altogether. We just keep the viral numbers down so that there are good active T cells that are able to fight off infections that would otherwise kill somebody with AIDS. I hope this was helpful. Stay tuned for the next lecture.